Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. So far we have set up the system for the hands on and we have learned basic Linux commands. So, we will utilize those commands today to look at the data and then we will do the quality control of a test data. So, let us start. So, these are the two concepts that we will be talking about today. So, we will talk about the data exploration, we will utilize some of the command line tools to look at some of the NGS data and then we will do a quality control on a FASTQ data set. Okay. So, these are the steps. So, the first step is the data download. Now, you might be doing your own experiment. So, you will get this data from the sequencing center or the company where you are doing your sequencing. So, you will need to simply put that data into your system, or if you are working on a server, you need to get that data to the server system. Okay. If you do not have your own data, do not worry. So, you can actually download some of the freely available data and you can test these pipelines, these commands yourself. Okay. So, I will give you some links here where you can actually download these data sets. Okay. These are part of some studies that have been deposited into these databases. And once we have the data set, we can look at the data, we can look at how the data looks and some of the aspects, and we will also learn little bit about the VI editor, how to use VI editor to navigate the data. Okay. It is not just for navigating FASTQ data, but you can utilize this for navigating any files or any data sets. So, there are certain commands with VI that makes it really convenient to use, especially if you are dealing with a really big data set. Okay. So, this would be the first step. We will look at some of the data and how you actually download this data. I will show you in a moment, and we will also then explore these data sets with VI editor. We will learn little bit about the commands that you can use with VI, and then we will actually do the quality control with a FASTQ data. So, for that we need the FASTQC tool that we have already discussed in the theory class, right. So, we will download this FASTQC tool, we will set this up and then we will do the data quality control. We will actually run our data through this FASTQC pipeline and finally, we will do the quality evaluation, means we will look at the output, right. And as we have discussed, FASTQC will generate certain statistics for you and we need to make sense of that statistics and we would have to make decisions ourselves, right. What we want to do before we actually go for downstream analysis. Do you need any pre-processing? Do you need any trimming? Those decisions would have to be taken by us. Okay. So, let us start. Let us look at some of the links where you can find these data sets, so that you can download, or if you have your own data, you can test all these commands with that data set. Okay. So, let us go to the links. Okay. So, here these are the links that I actually have shared with you. Okay. And this is the first one is an NCBI GEO, Gene Expression Omnibus database, where you can actually look at hundreds of data sets. Okay. So, there will be different types of data sets and you will see this kind of table when you open this site. Okay. Now, just to make sense of it little bit, right. So, here you see something called an accession number. So, this is a unique number that is given to each study. You will have a title of that data set, right, what this data set is about, what kind of study it is. Then you have something called a series type, right. This is whether this is an expression profiling, whether this is methylation profiling, what kind of experiment it is, right. So, this is again will specifically mention whether this is expression profiling. So, you can see this is expression profiling by high throughput sequencing, means you get transcriptomic data generated through NGS platforms. Then you have this column where you get the organism name. Okay. So, here it is human data set, but you can have other types of data set. For example, here you see mouse, you see Vibrio cholerae, etc., etc. Then you have a column with samples, right, and then of course, you have some contact, release date, when the data was submitted, etc. You can simply click on this accession, right, and you will see a lot more details about this data set. Okay. So, maybe we can zoom in a bit, right, and you can see. So, this is single cell RNA sequencing of ILCs and TL cells, right, and a lot of these details from this disease, etc. What is important? You see this is expression profiling by high throughput sequencing, and then you have a summary. Now, these summary files are sometimes very important because it gives you an idea of the study design, right, how this study was conducted. Okay. And before you do any analysis, you probably want to frame your questions, what kind of analysis you want to do, and then you utilize this data set. Okay. If you go down, you will see some of these ideas about these platforms. Right here, this platform is Illumina HiSeq 4000. Okay. So, we have talked about Illumina platforms, how it works, and then you have the sample names, right, and finally, you have the data here down here. Okay. So, we will look at this kind of data. This is a transcriptomic data when we actually do the transcriptomic analysis, when you go into the RNA-seq data analysis. You have other data sites, right, where you have this for example, European Nucleotide Archive. You can again look at some of the data sets that are here, or you can also look at something called Biostudies Array Express, where again you have similar kind of data, right. So, on the left you can actually choose which kind of data you are looking for, whether you are looking for methylation profiling by next generation sequencing, or maybe something else, right. So, this is something that again you can get. All right. So, once you have this data, right. So, let us now go to the terminal, right, where we can look at these data sets, right. So, here I will utilize one data set that I have already downloaded in the system, and we will do all the work on that data set. Okay. So, let us have a look. So, you probably remember these commands, the basic Linux commands that we have talked about, right. So, this is ls, it gives you the list of files that are present there, or you can use also ls minus l, it gives you a lot more information, right. On the left side you have seen, we have discussed already, these are the permissions of these files, and then we have the file names, the size of these files, right. You can see this, you can probably compare the size of these files. And what we are interested in are these two files. Okay. You have these read1 part dot fastq, and then you have read2 part dot fastq, right. These are the fastq files. Now, why do we have these names part, right. So, read1 read2 means this is paired end data, right. Immediately it is clear, rest of the name is same, right, but we have this read1 read2. So, this is paired end data, you have this information for two different reads, right. So, from sequencing from both directions. Okay. And then you have this part, this name part means this is a part of the original file. Okay. So, as we have discussed these fastq files, the data that is generated from Illumina platform, right, these are actually huge in size, okay, really big, because you have millions of reads in there. Now, if you want to do all these analysis with that kind of data set, will not be able to complete this hands on within a reasonable period of time, right, because all the steps they will take a long time to complete. So, that is why what I have done, I have taken only a part of the data set, right, so that we can do this analysis. I can show you all the steps in a reasonable amount of time, and the pipeline, the process remains exactly same, right. Whether you are dealing with 100,000 reads or millions of reads, the process does not change. Okay. It is just that it will take more time for the whole process to complete. Okay. So, we will work with these part data sets, okay, read1 part dot fastq and read2 part dot fastq. Okay. So, the first thing is we will actually use the VI editor to look at this data, and we will discuss certain commands that are very useful for navigating through these files. Okay. So, to open these files, right, we type either vim or vi, and we say the file name, okay, and VI will open it. Okay. So, now the file is open, you can have a look at the data, right. Now, one of the things I will warn you about is that do not try to open this with VI if you are working with a huge file, because that might actually lead to problems with the system, right. This might crash your system. So, I am opening this because I know this is a small file and I can show you some of the utilities of this VI commands. Okay. Now, this is FASTQ file, right, the extension you have seen. So, the first line is the header line, right. So, it contains information about the sequencing instrument, the run number, etc., the location of this cluster, etc., and then you have this one, and this is the read, right, read 1. So, this is 1, and then you have this filtering, etc. In the second line, you have the sequence itself, right, this starting with CSCC, right. This is the sequence of the read. The third line is the spacing, right, you have only plus sign, and the next line is the ASCII encoded quality score, right. So, this is the data, and this is repeated, right. This is one read, then you have the next read, right, and so on, and you can see we have about 10,000 sorry 100,000 lines here. You can see this right here, 100,000 L means 100,000 lines. Each read is taking 4 lines, means we have 25,000 reads. Okay. So, I have separated out 25,000 reads, and we will work with this 25,000 reads for all the analysis. All right. So, one of the things you probably notice is like it does not seem, the sequence does not seem to be in one line. Okay. So, it is because of the wrapping, right. So, text wrapping by the editor. Okay. So, if you want to remove this wrapping, there is a command in VI that you can use. Okay. Now, there are two things that you can do with VI. Okay. So, VI can be run in the read mode, which we are in right now. So, whenever you open a file in VI, it will be in this read mode. Okay. So, read mode means you cannot write anything, okay, and this read mode exists because we want to prevent any accidental writing on important files. Okay. So, this is the read mode, but you can enter the write mode when you are sure you want to modify certain parts, you want to add some data information, etc. Okay. Now, to enter this write mode, you have to press I. Okay. The moment you press I, what you get is this insert. Okay. You will see this here, right, all these details are here right at the bottom. So, you see this insert means you are in the write mode. If you want to come out of this write mode, you have to press escape, okay, and that insert command is gone. Okay. So, you can go into this write mode and you can start looking at this file. Now, in the read mode, you can run certain commands in VI. Okay. So, the one command that I mentioned is this, we do not want any wrapping around, right. We want this read sequence in one line. This is the format of FASTQ. So, how do you move this wrapping? So, this is the command, you can follow this here, right. I am writing these commands here, you can see is :SE, right, and then you have no wrap. We do not want wrapping. So, we just say no wrap, and you can see the format has changed. Okay. So, the sequence data now is in one line, then you have the spacing, then you have the ASCII encoded quality score. Okay. So, this is the actual format of FASTQ. Okay. And now, if you want to go to the end of the line, how do you actually go? It is simply shift. So, you press simply $. Okay. The moment you press $, you will go to the end of the line. Okay. And you can also come back to the beginning of the line by pressing this caret sign. Okay. So, probably know what is this caret sign. So, this caret sign will bring you back to the start of the line. So, these are really useful functions, right. You do not need to traverse through all these elements to get to the end of the line. You can very quickly go back and forth. Okay. Now, one of the questions you might have, can you go up and down very easily, right? And the answer is yes, we can. So, if you want to go to the end of the line, you can use these arrow keys, of course, but that will be cumbersome, right, if you have to traverse through the full file. There is a faster way to get to the end of the line actually. So, it is actually $. Okay. So, here we are, right, you can see we are at the end of the line. Now, on the right side here, if you see right at the bottom, you can see these numbers and this letter BOT bottom. Okay. So, this means we have reached the end of the file. Okay. And these two numbers here, the first number is 100,000, this is the number line number, okay, and this number 1 after this comma is the column number. Okay. So, you will see if we move to the right, you can follow the cursor here. If we are moving to the right, this number changes, right. We are moving into the these columns, right. So, this actually is counting the column number. Can we move to the top from the bottom? Yes, we can. We can simply say colon 1. We want to go to the first line. So, we can mention the line number and we will reach the first line. So, this works for all the lines. If you want to go to the 100th line, you can say 100, okay, and you go to the 100th line. Okay. So, these are really useful, right. You can see you can very easily traverse through these files without actually going through the whole file, right. Now, there are also other functions. You can search through these files very easily, like grep in the command line. We have seen how grep works, that is in the command line, but inside VI also you can search, okay, and this is how it works. Okay. You can simply press slash and you can give some text or numbers that you are looking for, and you will see, right, the moment I have typed, you can see this part is highlighted, okay, means it has found this number here, okay, 10224. We can press enter and it will, then if you want to search it again, you just press it again, right, and you will find another instance of this, and at the same time it will tell you, right, where it is finding these terms or these numbers. Okay. This is the line where it is finding, and this is the column, and it also gives you an idea where in the file you are. So, here is 31%, right, we have traversed 31% of the file, right. So, somewhere in the middle here we are finding these terms. Okay. So, that is a great thing, right. We can kind of search, we can traverse through these files very easily. Okay. So, let us go back to the first line, okay, and one of the things that you might be wondering is that if we can write anything on this file. Okay. So, to do that we have to go into the write mode. Okay. Now, before we go into the write mode, one of the things I will say, like if you are just interested in read mode, for example, with FASTQ files, we do not want to write anything, right. So, we just want to come out of that file. So, how do you come out? So, to come out of PI editor, we type colon Q. Okay. So, it will simply take you out of that file. It will close that file and you will come out of that file. Okay. So, this is very convenient, right. In the terminal you can go into certain file, you can open certain file, you can work on that file, you can come out of that file. Okay. So, how do you write here? Okay. So, this is something now we will discuss. Okay. So, let us remove this wrapping, right, and let us enter the write mode. Okay. So, here we are, we have entered the write mode now, okay, and let us say we write something like x y, something like that. Okay. We are just writing anything that we want. Okay. And so, once we are done, we can come out of that writing mode by pressing escape. Okay. Now, it turns out there are a lot of other functions that are very helpful for you for your work. Okay. So, for example, we have written something and we want to copy that multiple times. Okay. So, there is a shortcut in VI, right. In the read mode, you can simply press 2y twice, okay, and it will copy the line where your cursor is. Okay. So, we are in this line. So, if we press y twice, it will copy that line, and if you want to paste that line somewhere in the file again, right, we go to that line where we want to paste it, right, and we press P and that line will be pasted. Okay. Right. So, this is very convenient, is that we can copy this line very easily with these commands. Okay. So, the command is YYP. There is so this is kind of copy paste command, right. This is equivalent to copy paste in Windows. There is also cut paste. So, that works by saying DD, right, small d's, right, twice. If you press these, this line will be cut, and if you let us say want to paste it here somewhere, you can again say P, you can press P and then it will be pasted here. Okay. Now, this is copying one line or cutting one line, right. What about copying 40 lines, okay, or 50 lines, all right. So, how do you do that? So, the idea is very simple, right. So, if you want to copy let us say 40 lines, you simply say 40 y y, and you will see this here, it says 40 lines yanked, means 40 lines copied. Okay. And you want to paste those 40 lines here, maybe after this line you simply say P and it will paste those 40 lines here. Okay. So, this is 40 y y and then P, okay, P for pasting and y y is for copying. Now, you can do the similar thing for cut and paste, you can say 40 dd, it will cut 40 lines and it will paste 40 lines if you press P. If you do not press P, it will just simply delete those 40 lines. Okay. Now, if you say like we do not want to change FASTQ files, right. So, we want to undo all the changes that we have made. Okay. So, how do you do that? You simply say simply press U, right, and you can press U multiple times, right. It will go back to the oldest format, right, oldest form. As long as you have not exited the VI, right, all those changes will be removed, right, all those changes that you have made after opening the file will be removed if you press U. Okay. And then we can go out saying colon Q. Okay. Now, if you want to, if you are writing anything and you do not want to save it, right, let us say we again insert something by mistake, but we do not want to press U so many times, we want to just exit, but we do not want to save the changes that we have made in this file. Okay. So, how do you do that? We say colon Q and then exclamation mark. Okay. So, exclamation mark means quit without saving. Okay. Whatever I have written, that is by mistake, we do not want to save this. So, we just want to quit without saving it. Okay. So, we can go back and you will see this changes are gone, right, this SH, whatever I had written earlier, that is gone. Now, at times you want to probably make some changes and you want to save those changes, right. So, again we go into the insert mode, right, and we want to write something again, right. I am writing again something, but this time I want to save it. Okay. So, the command to save and to exit is colon W Q. Okay. And here we are out of that file. Okay. And if we go back again, we will see this line is there, okay, because we have exited by saving, right. So, this line will be there. All right. So, these are some of the basic commands that we can use. Of course, there are many more commands, many more functionalities, and that is why VI is really powerful, right. We can do a lot of things in the terminal just by using VI and opening the files, and there are commands within VI that work very well, and they can help us navigate these files very easily. Okay. We do not have to always traverse through the files or traverse through the lines. We can do a lot of things very easily using simple commands inside VI. Okay. So, I will delete that whatever I have written, because we do not, at the end we do not want to change the FASTQ file, and we will go to the terminal. Now, it is saying there was a change to if you do not want to save that change, add this exclamation mark, right. You see this command here. So, at times you get these kind of comments, right. If you are trying to do something that you do not, it is kind of like giving you a warning, right. So, that you do not make some mistake, right. So, we do not want to save this change, we want to save this change, right. We have removed this. So, you have to say colon W Q, right. We have removed that S Y whatever we had written. Okay. And now, here we are, right. Okay. So, this kind of summarizes, right, what you can do with VI, what you can kind of how you can navigate these files and modify files, add contents, right, write things, delete things, copy things, right, inside the file. Okay. So, that gives you a brief overview. So, now the next agenda is actually to look at the quality, okay, of these FASTQ files. Okay. So, to do that, we have these steps that we have talked about. The first is setting up the FASTQC. We will download and set up FASTQC, and then we will run FASTQC, right. We will generate this data that will be the quality data, right, and it will give us some idea about the quality of these data sets. Okay. So, let us start with that now. So, I will just clear the terminal and we will start with the FASTQC download. Okay. Now, installation of FastQC is actually very simple. If you just type FastQC in Google, the first page that you will get is the tool. Okay. So, you can simply click on that tool, right, and this is the page where you will be. Okay. So, here you can download this. Okay. Here is the link, download now, and you have all the instructions. You have readme, you have installation, you have everything in here. Okay. So, here what you have to do is just look at these two files. Okay. One is for Windows and Linux system, which is a zip file, the other is for Mac system. Okay. So, if you are working with Windows and Linux, download this file. If you are working with Mac, you download this file. Okay. So, I have actually downloaded this because it might take a bit of time for me to download. So, I have downloaded already, and I will show you how we can install and we can run this, okay, in the terminal. Okay. So, let us do that. So, here it is, right, here is the FastQC. Okay. Here is the FastQC, right, and we can extract this in here, right, inside this. Okay. So, here you have the copy of this FastQC. Maybe I will zoom in a bit, so that it's probably easier for you. Okay. All right. So, here is the FastQC zip file that you have downloaded, and we can simply extract this, right. And we can try also the command line, right, to extract this. Since you know the command line, try to utilize that. Okay. So, this is simply unzip, and let us see. Okay. So, this is down, this is okay. So, this is done, can clear, and we can simply say ls. Okay. And you will see this folder FastQC that has been created. Okay. So, this FastQC folder, right, inside this you will have that program. Okay. So, how do you go inside that folder? The command is cd, right. So, we move into that folder. Okay. And here you have a lot of these things, lot of files you can see. Okay. But do not worry, right, we do not have to worry about any of these things too much. The only file that we have to work with is this one, FastQC. This is the file that will execute, and this will generate the quality scores in the system. Okay. So, let us look at this permission of this file, okay, because if we do not have the execution permission, we will not be able to write this. Okay. Because sometimes when you download these files and extract, we might not have this execution permission, right. That is the only permission we need. We do not need read or write permission on that tool. Okay. So, let us look at this, right. So, how do you check permission? This is the command ls-l here, right, we are inside the folder. So, this will give you permission. Okay. So, this looks great, right. So, if you look at this FastQC file, we have read, write and execution permission, right. So, we can execute this code, we do not have to worry about anything here. Okay. So, let us now execute, okay, and see how we generate the quality. Okay. So, there are two ways you can run FastQC in Windows system especially, right. So, in Windows system you can run it just clicking on that FastQC inside that folder, right. If you have Java installed, it will simply open up a window, right, and you can load a FastQ file and you can generate all the quality statistics. There is a better way if you are running especially WSL inside Windows, you can run FastQC through the command line itself. Okay. And in Mac also you will run through the command line. So, what is the advantage of running through command line? Because if you have a lot of data and you want to do quality check of all of those, okay, it is better to run through command line because you can run using a single command. If you are running through that program window, you would have to load individual files, right. You can do only one file at once, and you will have to do this repeatedly. For if you have, let us say 10 files, you have to do this 10 times. So, we will prefer using the command line route. Okay. So, let us utilize this command line tool and then do the quality check. Okay. So, how do you call this program FastQC inside this folder? Okay. So, to run that I will just come out because our data file are here in this folder, right. So, we need to give these data files to that program. Okay. So, I will now call this program, right, because this is inside this folder. So, I will have to go inside that folder to call this program. Okay. And you notice this is dot, right, I have given this dot slash, means this is we are executing this program, right, we are calling this program in there. Okay. So, we will this is how we will call this program, right. So, and then we would have to give the names of the files on which we want to run this quality control. Okay. So, here these are the files we have, and you can specify multiple files, okay, one after another. Okay. So, we have two files, we will run them here, we will mention this read one part dot first q and then followed by read two part first q. So, what you will see, first QC will run this quality control one after another. Okay. So, if you have 10 files, it will run this for 10 files consecutively. Okay. So, we are running this and it gives you an update, right, what percentage of run is complete, etc., right, for each of these reads, right. So, once it is done, right, so it will give you this it will be back here, right. So, this is done now, okay, and we can now go and check the quality score. Okay. Okay. So, let us go inside that. So, you will see, right, inside this folder you have these 4 files will be created. We have run 2 first q files. So, for each there will be 2 files generated by first q. Okay. So, you have this read one part first QC, right, this is HTML and then you have the zip file. Okay. This will be clear if we actually do this in the terminal. Okay. So, let us do this in the terminal and you will probably see this clearly. Okay. So, you can see this first QC dot HTML, okay, this has been created, and for read one this is another file that has been created, right, part first QC dot zip. So, all the results are inside these 2 files. Okay. And then you have similarly for read 2 you have these 2 files created. Okay. You have read to part first QC dot HTML and read to part first QC dot zip. Okay. So, what we will do is I will create a folder saying results first QC, right. So, we can move all the first QC results inside that folder and we can then check, right, the results and make decisions. Okay. To create this folder or directory is mkdir in Linux, right, and we will move these first QC files, right. So, we want to move these first QC files, right, and the command is mv, right, move. Okay. So, we have this part first QC star and then the destination directory, right, where we want to move. Okay. Hopefully, you remember from the basic commands that we discussed. Okay. And similarly, we have done this for read 1, we will do this for read 2 as well. Okay. And you might be wondering, right, why I am giving this file name dot star. Okay. So, star is a wildcard, means it will match anything it will find. Okay. So, at once it will move this HTML as well as zip inside this folder. Okay. So, this is moving it, and now we can check, right, results first QC. You see all the first QC results are inside this folder. We can move now inside this folder and we can unzip, right, we can simply say unzip the zip folder so that we can check the results. Okay. And we do this for both of them, right, for read 1 and read 2, and we have now all the results created. You can see these 2 folders now created, these are unzipped ones, and inside this you will have the results. Okay. Now, we can go inside this and we can do the quality evaluation and we can make our decisions. Okay. So, in the next class we will actually look at this data, right, we will look at this folder, we will look at all the results that have been generated, and then we will do we will make decisions and we will do the pre-processing. Okay. So, with that I will stop. Thank you.